everyone, my name is Valerie and I'm the Applications Manager at RedShiftBio. Today I'll be going through our application note on how we can detect pressure-induced protein aggregates and distinguish those aggregates from natively folded beta sheet structure using MMS. If you want to read the full app note, you can find it on our website. But first, I'd like to introduce RedShiftBio and our instruments, the AQS3 Pro, which is our first generation tool, and our second generation tool, the Apollo, which is pictured here. RedShiftBio is the inventor of Microfluidic Modulation Spectroscopy, also known as MMS, which is the technology platform used by both our first and second generation instruments. These tools are used for sensitively determining protein structure using a high-power quantum cascade IR laser. The study we'll walk through today focuses on immunoglobulin G, or IgG, and commonly you'll see antibodies like this depicted as a simplified cartoon where you can see the heavy and the light chains clearly distinguished by color, in this case green and blue, and the Fab region that is unique with the variable antigen binding domain. However, on a molecular level, the structure is more complicated and actually mostly made up of antiparallel beta sheet secondary structure, as seen in this figure. Since protein structure is closely linked to its function, structure information is highly valuable as one of the critical quality attributes. In this case of antibodies, this means that structure will dictate its antigen binding ability, and lastly, the structure will impact the protein's stability and influence its final formulation and storage conditions. Pictured here is a crystal structure of an amyloid beta protein fibril, similar to those that make up plaques in Alzheimer's patients. This is made up of multiple amyloid beta polypeptide chains, where each different color is an amyloid beta monomer. If we follow just one of the polypeptide chains and look top down, you can see these fibrils are stacks of antiparallel beta sheet structures. Misfolded protein can lead to exposing of hydrophobic pockets that cause further aggregation. Aggregates are a problem because they are non-functional, can be immunogenic and disease-causing, and are usually beta sheet rich, which, as we saw in the previous slide, so are natively folded antibodies. Since we've just seen how protein structure is critically important to its function, it is important to be able to distinguish protein aggregates from natively folded protein, since they can both be made up of antiparallel beta sheet structure. So our research question is, with the increased sensitivity of MMS, are we able to distinguish native from aggregated beta sheet structure? And if so, how low can we go in terms of quantitatively detecting small amounts of aggregates? To answer this question, we partnered with Pressure Biosciences and used their barocycler to create stable soluble aggregates. We did this by stressing a sample of IgG at 60,000 psi. The tube on the right is the pressure-treated IgG, and the one on the left is the untreated sample. Then, using the stressed sample as a surrogate for misfolded aggregates, we spiked in small amounts of the treated sample into the untreated sample at increasing amounts, and used sample 1 as the untreated control and sample 11 as the 100% treated control. Each of these samples in the table were then run on our MMS system to quantify the structure. The absolute absorbance graph here shows native IgG, where the main peak is centered at 1640 wave numbers. As we spike in the treated sample, the center peak decreases and the region at 1620 increases. It is well understood that the region at 1640 in the IR spectrum is due to native beta sheet structure, and 1620 is due to aggregated beta sheet structure. From here, the software automatically calculates the second derivative of the absolute absorbance plot in order to enhance regions of change. Taking it one step further, we can also select a control. In this case, it's the 0% spike sample. We subtract that sample from all the other samples, which leaves us with a flat line for the control, and anywhere that deviates is a region of change. Now we can see very specifically where the most change is occurring. By tracking specific wave numbers in the delta plot that we know are important for secondary structural features, we can track the changes to those structures across the whole sample set, Shown on the right is what we call the stability plot and is monitoring 4 wave numbers of interest, specifically 1624 and 1697 for aggregated beta sheet structure and 1640 for native beta sheet. We also track 1656 for alpha helix, but as antibodies have very little alpha helical content, this one's not important to focus on. If we do focus on one region, specifically the intermolecular aggregated beta sheet structure at 1624, and fit this to a line of best fit, you will see the excellent linearity across the data set with an R-squared of above 0.999. We can use this plot to calculate the limit of quantitation, or LOQ, which is the lowest amount of aggregates that can be accurately detected beyond the noise. In this case, we're able to accurately detect about 5% aggregated spiked. Lastly, the software also calculates the percent higher order structure and presents it in a bar graph here. This corroborates and quantitates the decrease in native beta sheet structure and increasing aggregated beta sheet, denoted by the beta minus. Overall today, we showed in this app note that MMS is able to differentiate the 2 types of beta sheet structure, the native and the aggregated. With the spike series, we showed that MMS response is highly linear and quantifiable. Lastly, we demonstrated that the HOS corroborates the loss in native beta sheet structure and the gain of aggregated beta sheet structure due to the pressure, stress, and spiked study. Thank you so much for listening. I hope this was interesting, and if you want to read the full app note, you can find it on our website under the Resources tab. Thank you.